Hello, welcome along. Good afternoon. My name is Mike, and I'm a lab engineer here at BioReperia, and I'll be your moderator for today's session. So today's webinar, the latest in our series, is co-hosted by both us at BioReperia and InVivo Biosystems. The webinar today is Advancing Human Disease Research with Zebrafish Models from Tank to Bedside with Rapid In-Vivo Analysis. The webinar will run for approximately 45 minutes, and we will have two speakers, and then we'll have a brief Q&A session. A couple of things first. One, if I could ask you to turn your microphones and cameras off, that helps that the presentation runs a bit smoother. Also, our webinar will be recorded, and we will later upload it to our YouTube channel, so you'll be able to share it with your friends and your colleagues and watch it back later. So, as I said, we will have two speakers, and now I will make the introductions. Our first speaker is Dr. Joseph Bruckner. He's a neuroscientist by training, and he received his PhD in cell and molecular biology from the University of Wisconsin-Madison. And he studied how gut microbes influence brain development in zebrafish. As a postdoctoral fellow in the laboratory of Judith Elson at the University of Oregon. As director of biology at InVivo Biosystems, Joseph creates zebrafish and C. elegans models for identifying new therapeutics for diverse, underserved conditions. Our second speaker will be Dr. Gabriela Vazquez. She is a project manager here at BioReperia. She has a master's and a PhD in molecular biology and biotechnology, and she did her postdoc here in Sweden in Linköping University, where she studied the molecular mechanisms for tumor microenvironment that drive breast cancer progression, angiogenesis, and metastasis using different techniques, which include spheroid cultures and the zebrafish xenograft tumor model. She has accumulated more than 10 years of experience working with zebrafish as a cancer model. So, if you could just go to the next slide, someone. The webinar outline. So, like I said, we will start with InVivo Biosystems, and there'll be first an overview of zebrafish as a human disease model, their Rapid Gen platform, and the preclinical development case study. And that will be followed then by BioReperia, who will outline the ZTX platform and finish with a summary. So now I will hand you over to Dr. Joseph Bruckner. So if you want to take control and take it away.
Yeah, thanks for the introduction, Mike. It's a pleasure to be here and to speak to everyone. So I wanted to start by telling you a little bit about who we are at InVivo Biosystems. So here's a few interesting facts about us as a company, but we specialize in creating custom genetic models that can be used to identify therapeutics, but also for fundamental, the basic science, mechanistic research. And we're really good at this because we've got experienced staff that we've gathered a number of zebrafish experts over our time as a company, and that enables us to generate really powerful results for our partners. You can go to the next slide. And so before we jump into some of the advantages of zebrafish as a model system, I wanted to emphasize that zebrafish are increasingly recognized by the broader research community and the pharmaceutical and biotech industry for its power in modeling human diseases. So this is evident in a number of metrics, so not just the number of publications that increase year over year using zebrafish to model human diseases, but also that drugs have been developed using zebrafish research and are now have entered into clinical trials and are even used in the clinic. I also wanted to call out that some of the history of zebrafish research and zebrafish as an international model system. So it's started back in the 80s, founded initially by George Streisinger at the University of Oregon. This is a picture of George here on the left. And InVivo Biosystems is as a company spun out of the University of Oregon. And we're still really proud to call Eugene, Oregon, our home. It's where our offices and lab space is located. And we continue to really closely collaborate with the University of Oregon, which is a big strength for our platform that we can use not only their physical resources, but we have access to a wide body of zebrafish research experts that continue to work at the University of Oregon. You can go to the next slide. So zebrafish as a model for human diseases have a number of significant advantages. I wanted to highlight a few of them here, but know that this is a small selection. So one that we talk about a lot on the right is that the embryos are transparent, and they continue to be largely transparent throughout a significant chunk of their development, which is very rapid. So the entire body plan in zebrafish is laid down within a matter of days. All of the most of the organ systems that humans have are developed in that time, and it's very easy for us to do experiments on intact animals because of this transparency and rapid development. This is part of what makes them a really great ethical choice for drug development research, because it means that a lot of our methods can be less invasive and therefore induce less pain and stress, on top of the ease of using the models relative to rodents. And another sometimes underappreciated aspect of that is the ex utero development. So the fact that all of this early development is happening outside of mom, in contrary to mammals, is a big part of what makes it very easy to work with these animals and what makes them powerful as a model system. The last thing I wanted to call out is that we have, in part because of the ease of access to the embryo, really great genome engineering techniques. And so in many cases, we can do gene editing. We have a suite of gene editing capabilities that is comparable to rodents, but can also do those techniques in a high throughput manner, including some gene humanization. You can go to the next slide. So all of this means that a number of human diseases are gathering are getting benefit from studies in zebrafish, and here I wanted to highlight those that we and our partners at BioReperia work with frequently. So that's diseases such as immunological disorders. We have a significant focus on rare monogenic diseases as well. That's tied to our abilities to do genome editing in zebrafish. We also focus a lot on neurological indications, and a big advantage here is what I was mentioning earlier about the transparency of the embryo, so we can visualize the brain in intact animals as it's developing. We're also able to address any number of kind of organ or tissue level disorders using a combination of the experimental ease and the ease of making genetic models. So we can, label kidney cells, and it's very easy to then visualize and manipulate them, as an example. And then later in this webinar, Gabby will speak a lot to their utility as a model for cancer. Go to the next slide. So I wanted to give folks who aren't familiar with the model a bit of a brief introduction to zebrafish life stages, and kind of what the animals look like and how we work with them. So on the left here, you can see that from a single mating pair, we can generate a very large number of offspring. So this large brood size, up to potentially 300 embryos from a single pair, is part of what enables the high throughput experiments in this model, as opposed to, a handful of pups from a mating pair of mice. And then the easily accessible rapid development that I was talking to is visualized here in the center. You can see that in a matter of hours, embryos divide, and we have access to the dividing embryo at all of these stages. So starting just after fertilization, we can inject single cell embryos with any number of things, but this is typically the point at which we're modifying the genome to make stable transgenic animals. And then by starting in the first day of life and through the first week of life, like I mentioned earlier, the animal is starting to lay down all of the body systems that'll persist into adulthood and all of the body systems that are present in a mammal as well. So you can see it's starting to look a lot more like a fish by a week old. It's got these animals are mostly eyes at that stage, but it's got a well-developed brain, vasculature, heart, immune system, things like that. And then by 3 months old, the animals are sexually mature and considered breeding adults. So at InVivo Biosystems, we do experiments on all these life stages, though our preference is often to work with these larval stages because of the ease of accessibility to many different organs and tissues. So the last thing I'll say on this slide is that, example of this is that we can take individual larval animals and put them into microplates, and because of their small size, we can do a number of analyses, and one that I'll talk about a lot later is we can track and analyze their locomotion as they swim around in these individual wells. But we do a lot of other assays in a microplate format, things like plate reader, such as plate reader assays. You can go to the next slide. So I wanted to give everyone also a brief overview of InVivo Biosystems RapidGen platform, because I'm going to focus on a few very specific indications and examples of work that we've done with our partners. But I wanted to make sure that you knew that our capabilities have a pretty broad reach. So many of our partners want to be discovering either novel compounds or repurposing existing drugs or biologics. And through our platform, we're able to help them prioritize those lead compounds and then provide validation of efficacy in either just zebrafish or in multiple model systems. So wanted to call out that our specialty is alternative models. So at InVivo, we work not only with zebrafish, but also with C. elegans. And we have cell culture and induced pluripotent stem cell capabilities through our partner, Scintillant. And then our platform has a number of patented technologies that we can apply to those models, everything from CRISPR, knockouts or knock-ins, chemical treatments, gene humanization or protein stability studies. And then, of course, through our partner BioReperia, you'll hear a lot about how we can also do xenograft studies. And then we can use that combinations of models and technologies to address a number of diverse indications for our clients. So that's everything from neurological to cardiologic indications. Go to the next slide. So I wanted to give you a couple examples of how this might look for our in our existing partnerships. So one example is that we could start with optimizing CRISPR reagents, injecting early embryos. That's what you see here at the top. And then confirming that those embryos within a week have the expected phenotype. So that could be either via microscopy, like I was speaking to, or on the bottom here, we're representing that high throughput movement assay. And then the advantage of this, of the ease of working with zebrafish, is that we can also apply a lot of molecular techniques. So we can take those animals straight from that cellular or organ level phenotyping and extract RNA and via RNA sequencing, do gene expression analysis for our clients that provides not only this efficacy data, but mechanistic insight into the efficacy of their lead compounds. We can go to the next slide. It's also worth noting that this can be applied, again, via our partners at BioReperia, to even to samples of mixed species. So you'll hear a lot about this technology from Gabby, but we can do a similar sort of RNA sequencing analysis on animals with a human tumor xenograft, where we can take those intact animals, homogenize them, extract RNA, just like I was talking about before, and then through our partnership with the sequencing, or the, sorry, RNA sequencing analysis platform, Rosalind, we can separately analyze the differential expression of zebrafish genes and human genes in the same animal to assay specific drug effects on each tissue. And a lot of our partners really enjoy the fact that we use the Rosalind platform for this, because this enables us to give them direct access to their data in Rosalind, so that they can not only see our interpretation of what the effects of their drug is, but they can play with the data themselves and be involved in the discovery in that extent. Next slide. So I wanted to give you another one more specific example of how our Rapid Gen platform has worked for a partner. So in this case, we started with a zebrafish Rapid Gen model. We applied our CRISPR technologies to address a neurological indication. Go to the next one. So in this case, we optimized CRISPR reagents to target the genetic disorder that our partner was interested in. I'll tell you a little bit more about that approach in a second. Injected single cell embryos, and then within a week, we're doing movement analysis on those F0 animals, and here we were tracking movement that is specifically related to seizures, because the client was interested in modeling a seizure disorder called Dravet syndrome. We then applied drugs to this model, after having confirmed the seizure-related movement phenotype. Oops, my lights turned off. And assayed whether their proprietary compounds were able to restore this seizure-related phenotype in the genetic model that we created. Go to the next one. So here's a little bit of technical detail about how we created this genetic model. A big advantage here, like I mentioned, is that we're working within the first week in an F0 animal, rather than having to wait months, potentially years, for a stable mutant line. So we did that using a technique optimizing the genetic reagents to disrupt the gene in multiple different exons. And by injecting these reagents that combine to have a really high level of loss of function of this gene, we can be confident that we're overcoming the mosaicism that you see when you inject a single cell animal, typically with CRISPR reagents. So this results in F0s that, when they're 7 days old, 70 to 80% of the cells in every single fish will have a loss of function of this gene. So this is the gene SCN1 lab in zebrafish. And we can analyze the degree of loss of function. On the right here, you're seeing an example of Sanger sequencing traces that we can deconvolve to get that math and determine the degree of loss of function. It also helps that these animals have obvious morphological phenotypes. So you can see the mutant animals on the bottom here They are hyperpigmented, so they're really dark, and they don't properly inflate their swim bladders. Next slide. So the assay that we then ran on these zebrafish, like I mentioned, this is a zoomed in image of the 96 well plate where each well has an individual zebrafish and we can track their movement over time. So I'm showing you example traces of that tracking. And then also in this behavior chamber, we can apply different stimuli to the animals. So in this case, we applied a light stimulus where they start out moving in the dark, which zebrafish actually typically prefer. Then we turn the lights on. And when that happens, normal zebrafish, so you can see the injection control or the wild type, the red or black lines in this graph, normally they have a really mild startle response to that change in the lights. But what I hope is obvious is that our SCN1 lab F0 mutants have a really exaggerated startle response. And in this model, this is thought to have to do what this is thought to be a seizure-related movement. And so you can see I put an example of our quantification of this phenotype on the bottom. So again, you can see when we measure the burst in swimming that these animals have in response to this light stimulus, the wild type or injection control burst is very small. In purple, the SCN1 lab mutants have an exaggerated burst. And then we exposed these mutant animals to 2 different concentrations of our client's proprietary compound, and which you can see had a dose-dependent effect. So in the 2 blue bars, that especially at the highest dose, that this compound was able to partially rescue this seizure-related phenotype. This was in comparison to a control that we often use in this particular model called fenfluramine. So fenfluramine is a FDA-approved drug that's used in the clinic for Dravet syndrome. And so we were using this as a benchmark for restoration of seizure-related activity in this model. I think that is all I've got. I think I'll hand it over to Gabby now.
Yes, thank you very much, Joseph. So then I will take over with regarding the zebrafish as an oncology model. So why do we use the zebrafish as a human avatar to model cancer disease? Well, this is because as Joseph mentioned, the zebrafish model has so many advantages. But in regards to, well, added to that, in regards to model cancer, human cancer disease, it includes different benefits, such as it has a high accuracy in predicting drug response, as it's observed in the clinic, and also the dissemination as, and it serves as a prognosis tool to say if the patient is going to progress to metastatic disease. It's also possible to evaluate combination of treatments, for example, several standard of care treatments for different indications. There are 2 or more chemotherapy drugs that are used, so it's possible to evaluate in the zebrafish embryos and the zebrafish model. The embryo stage, the model is fast. It's only in 3 days. It's possible to have results on primary tumor and metastasis. It's also a low cost, being that they are very small and they can be housed in very small tanks, and the embryos can be evaluated by using multi-well plates. It's very amenable for screening different compounds or different cancer indications as well. And since in the zebrafish embryo can be faithfully recapitulate the tumor microenvironment, so it's highly suitable for immuno-oncology analysis. And besides, it has an ethical benefit because at the embryo stage, the zebrafish are not considered animals, so there is no need for an ethical application for working with the zebrafish embryos. Of course, as any preclinical animal, it comes, preclinical in vivo model, it comes with its limitations. For example, the short assay time of only 3 days. And this, for several people can think, for example, immune checkpoint inhibitors that take some time in the mice for evaluation of this type of drugs. However, as I'm going to show in later slides, this is possible to do for the zebrafish embryo model. However, for other assays, such as the resistance studies or dose escalation studies that require these different doses at different times, then for that it's difficult to do it. Of course, pharmacokinetics, because of the at this embryo stage, we are missing some organs as well, and this is and a different limitation is the small tumor size. So for this is challenging to perform a post-evaluation study. However, it's still possible, as I will going to show it later on. And we have an early treatment onset after implantation. So as I'm going to show you later about how this zebrafish tumor xenograph model works, the treatment, it's applied right after implantation. So we don't have that window where we let the tumor grow before treatment starts. So yes, so how this zebrafish tumor xenograph model works for both in cancer research, but also later on in precision medicine. Well, everything starts with, I will just change here to the laser point. Everything starts with human cells, tumor cell suspension that can come from a cell line or a PDX model or a clinical sample. This cell suspension is labeled with the red fluorescent dye and then implanted subcutaneously, as we can see here in this image, in 2 days old zebrafish embryos. So after implantation, these zebrafish embryos are placed in a multi-well plate, 1 embryo per well, so we can follow each embryo throughout the evaluation time independently. So after the tumor-bearing embryos are selected for correct implantation, so the treatment can be added to water easily if we are talking about the small molecules like chemotherapeutic treatment. But if we are talking about larger molecules like peptides or antibodies, ADCs, those can be mixed together with the tumor cells and implanted together with the tumor cells, or can be injected intravenously after tumor implantation. So after 3 days after treatment, the zebrafish larvae can be imaged for in the primary tumor and also in the tail for metastatic evaluation. So with this model, we can have 2 different outputs. One is on tumor regression, and the second one is on metastatic dissemination. There are different administrations that we can perform using this zebrafish embryo model. For solid tumors, it's more commonly used subcutaneous implantation in the perivitalizing space. So after 3 days, we can evaluate primary tumor regression and local dissemination as well, and the metastatic dissemination in the embryo tail. For blood cancers or liquid cancers like leukemia cells, this is more commonly administered intravenously into the zebrafish embryos. So what's evaluated after 3 days is just the capability of these cells, leukemic cells, to establish metastasis or the metastatic lesions in the fish after this evolution period. And for later on for cancer progression and the metastasis, where the zebrafish xenograph models can provide also unique advantage in this cancer research because of their optical transparency, as Joseph was mentioning, and their genetic adaptability. So the zebrafish can be genetically manipulated to create these transgenic cell lines, such as the one shown in this slide, expressing enhanced fluorescent protein in the endothelial cells. So this genetic feature allows for this clear visualization of the embryo's vascular network, and facilitates the analysis of the, for example, tumor angiogenesis in the primary tumor, as we can see here in this image. So the rapid development of the zebrafish embryo also allows for the tumor xenografts to establish a unique tumor microenvironment with all these intratumoral blood vessels that can support cancer cell dissemination. In this other video or image, we can see that the disseminated cancer cells commonly establish metastases within a region called the caudal hematopoietic plexus in the zebrafish embryo, where we can evaluate the interactions between the tumor and the immune cells, and that can be observed in real time, as demonstrated in this time-lapse video. I will just click here. So as you can observe, we can see the cancer cells labeled in red, and the immune cells, in this case neutrophils, labeled in blue. And both cell types co-migrate to the metastatic niche, where the cancer cells are observed moving through the highly vascularized tissue alongside with immune cells, utilizing these immune cells for transendothelial migration within the metastatic niche. So overall, this data shows that the transparency of the zebrafish embryo allows for this real-time observation of the mechanisms that mediate cancer cell dissemination, enabling also the development of potential therapies targeting early steps in the cancer metastasis process. Yes. And in this slide, presents two time-lapse videos captured over a 12-hour period following the subcutaneous implantation of breast tumor cells in red and immune cells in blue. And these immune cells are, again, the neutrophils that were co-implanted. And the video in the left, we observe that breast cancer cells actively recruit the immune cells to facilitate the invasion into surrounding tissues and subsequent intravasation, enabling dissemination throughout the embryo. So this close interaction between the breast cancer cells and the immune cells lead to the co-migration from the primary tumor site. So by the end of the video, as you can see here, a significant proportion of breast cancer cells have disappeared, have metastasized. So in contrast, in the video here on the right, it demonstrates that by using immune therapy-mediated inhibition of these cell interactions between the tumor cells and the immune cells can effectively suppress the immune cell-dependent cancer cell invasion and dissemination, as we can observe here that the tumor cells are barely migrating in the zebrafish embryo. Later, to investigate the mechanisms that drive breast cancer cell invasion and metastasis, we examine the role of the estradiol, since we are talking about breast cancer model here. So the estradiol has a very important effect on breast cancer, especially for cancer cell lines that have high expression of the estrogen receptor, such as the MCF7 cells used in this study. So we evaluated the effect of estradiol in promoting the protumoral polarization of immune cells, specifically for, in this case, the neutrophils. And we evaluated the dissemination of the low metastatic breast cancer cells, which is these MCF7 cells, under conditions of estradiol treatment, both in the presence and absence of human neutrophils. So our findings indicate that while the estradiol alone did not affect breast cancer cell metastasis, it is affected when the neutrophils are co-implanted with the tumor cells. So the neutrophils increase significantly the dissemination of the low metastatic tumor cells. And this effect was further amplified when estradiol was present. Further analysis revealed that estradiol enhances the TGF-beta production and secretion by the cancer cells, which in turn upregulates the expression of the lymphocyte function-associated antigen integrin, or LFA1, on human neutrophils, which is a critical integrin for neutrophil diapedesis and cell-to-cell interactions. So immune therapy targeting either TGF-beta or ILF1 integrin in the zebrafish model resulted in a significant inhibition of the neutrophil-mediated breast cancer cell metastasis. So our findings from this and other studies indicate that tumors exhibit distinct invasion mechanisms depending on the cancer type that it's implanted, with each tumor's invasive behavior in the zebrafish model correlating with what we observed in the clinical, in the case as it is observed in the clinic. So this data highlights the use of the zebrafish model for investigating the tumor microenvironment interactions that can drive tumor progression and metastasis. And also it offers insights into the potential therapeutic targets, as shown in here for the LFA1 integrin, and supports for the development of drugs that aim at modifying the tumor microenvironment to inhibit tumor progression. Also, the zebrafish tumor xenograft model can be used, as I mentioned before, for immune oncology. And in this case, in this experiment, we evaluated the immune checkpoint inhibitor, the anti-PD-1 antibody. So for this study, what we performed was we used melanoma tumor cells, which were labeled with this red fluorescent dye and implanted subcutaneously. And we used matched tumor infiltrating lymphocytes, which were labeled in red. Sorry, in this case, the tumor cells were labeled in blue, and the tumor infiltrating lymphocytes were labeled in red. And these lymphocytes were administered or injected IV right after primary tumor implantation. So with or without the anti-PD-1 treatment. So after 3 days, we evaluated tumor size and also infiltration of these TILs into the primary tumor, and also the metastatic dissemination. What you can observe here in the first graph here in the upper panel, it's the primary tumor size that we observe after 24, 48, and 72 hours after treatment. And in here we can observe that the TILs, they have a cytotoxic activity by themselves. So they, the presence of TILs, as we can see here in the third graph, third bar, we can see that it significantly decreases the primary tumor size compared to the controls that we have here in this study. So, and this cytotoxic activity, it's further what's further enhancing presence of this anti-PD-1 antibody. Here in this panel, you can observe the different example pictures for embryos in control group, or in presence of just the anti-PD-1 without any presence of the TILs. When we have the presence of TILs, we can start observing that these lymphocytes start infiltrating into the primary tumor, and this infiltration of TILs in the primary tumor, it's further enhanced in presence of the anti-PD-1 treatment. And we can observe also this reduction of the primary tumor size as well. This same data, you can see it here in this graph, where we can, after 3 days, we can see a significant increase in the infiltration of tumor lymphocytes into the primary tumors. So the zebrafish model, the embryos can it's possible to use the embryos to evaluate different PDX models and to evaluate the correlation as well that we have with the mice PDX models. So this study was performed in collaboration with Charles River. So at Charles River, they received their clinical sample, and the clinical sample they expanded, they implanted in mice, and they expanded. So they create a vast library of PDX models. So these PDX models, we received approximately 30 different lung cancer PDX models from Charles River. So we received the two more pieces, the two more chunks cryopreserved. So what we did in this study is that these 30 models, these tissues were dissociated mechanically and enzymatically, labeled as previously shown with our fluorescent dye, and implanted subcutaneously into the zebrafish embryos. And the embryos were treated with erlotinib and paclitaxel. So out of the 30 different models that we implanted, 25 allowed for the generation of sufficiently large number of tumor-bearing embryos, showing a take rate ratio of 83%. So by using this, the zebrafish model, we could detect those PDX models that were responders, shown here in green, and non-responders, both to erlotinib and paclitaxel treatment. And we can say that by comparing how it went for the zebrafish model compared to what was observed with the mice, we can say that we have an 87% of the models that exhibited the same treatment response towards erlotinib and paclitaxel as it was generated in the PDX, in the mouse PDX models. Yes, so this zebrafish model, embryo model, can also be used for precision medicine, which is quite interesting. And it, as mentioned before, since they have a highly translatability prediction or high prediction and of drug responses in patients. So what you can see here is the results of several clinical trials that we have performed for various indications. In here you can see the results for urinary bladder cancer, which we treated the embryos with a combination of treatment and MVAC, that is just in the clinic, and we observe an 80% prediction of treatment outcome as observed with the patients, and 100% sensitivity when predicting a lymph node or dissemination, metastatic dissemination with patients. And similarly, we observed that with the epithelial ovarian cancer. So, and this high accuracy and high translatability of the zebrafish model is supported by other labs, which they have published their different results in different journals, different publications, where they observe that the zebrafish tumor xenograft or zebrafish avatars can show different accuracy levels from 80%, as we showed, and 100%. But the only difference is that the number of patients here that goes from 2 to 6, and this obtained data is from about 50 different patients. So following in that same line about the prediction of drug response in patients using the zebrafish tumor xenografts, and here I'm showing you a clinical study that was conducted to assess whether the zebrafish avatars could benefit the effectively predict clinical outcomes and impact the time to recurrence in cancer patients. The tumor samples from patients were implanted into the zebrafish avatars, which were then treated with the same therapies as the patients themselves. And this approach, using this approach, successfully generated zebrafish avatars for 55 out of the 79 patients. having a 70% success rate. And the study included patients diagnosed with colorectal cancer, renal cancer, and liver metastasis across stages 2, 3, and 4. So the progression-free survival was evaluated using these Kaplan-Meier curves, comparing outcomes for patients with Zebrafish avatars that were responsive or sensitive versus unresponsive or resistant to the treatment. So the progression-free survival was measured from the start of chemotherapy to the last recorded follow-up, or the date of disease progression. So the results show that patients with a sensitive avatar had a significantly longer average of progression-free survival of 30.9 months compared to 7.9 months in those with resistant avatars when analyzing. And then when analyzing by the cancer stage 2 and 3, we saw the same response. So as a summary, well, the Zebrafish avatars in this study demonstrated 91% positive predictive value, which means 91% of the time detected those that were sensitive, and 90% of the time detected those patients that were resistant to the treatment. So yeah, after primary tumor and metastasis evaluation in the Zebrafish embryo, different post-evaluation assays can be performed to gain a deeper understanding of the biological processes that drive the cancer progression, survival, and resistance in response to various stimuli, such as drugs and immune challenges, environmental challenges, or different stressors. So for this purpose, the Zebrafish embryos can be preserved in a variety of solutions. For example, RNA later that can be used to preserve tumor-bearing embryos for further RNA-seq analysis and perform comparative transcriptomic analysis, as Joseph was mentioning earlier, between the cell line-derived xenografts, as is seen in this example figure, to evaluate gene enrichment in different biological processes, such as immune response, metabolism, signaling, or genes involved in the epithelial to mesenchymal transition, proliferation or hypoxia, and also angiogenesis. Another thing that can be done is lyse the embryo-centripetal buffer to generate lysates for protein biomarkers and evaluate this by ELISA or multiplex assays. So the tumor-bearing embryos can also be preserved in 4% PFA to perform whole mount staining or paraffin embedding of the embryos for immunofluorescence staining or immunohistochemistry for the primary tumor xenografts to evaluate specific outputs and markers, for example, proliferation, apoptosis, tumor angiogenesis, among others. And this can also be combined with a confocal microscopy to have a single cell resolution and the 3D architecture of the primary tumor xenograft. And also enzymatic dissociation of the tumor-bearing embryos can also be performed to separate the tumor cells from the embryos by flow cytometry and perform single cell sequencing to help researchers to understand the complex biological processes that a single cell resolution can reveal by gene expression patterns that can contribute to aggressive behavior or the metastatic potential in the individual cancer cells. So yes, as a summary, we have that the Zebrafish model can be used for many different evaluation and as a model as different human diseases, not only in cancer research, but thanks to the high homology in disease-related genes with humans, it's amenable for modeling for different diseases, for high throughput screening compounds, being that they are very small and at the embryo stage, and they are quick and easy genetic manipulation. And there is an easy drug exposure as well for the toxicity testing. And the latest is due to their high translatability, can be used for precision medicine, as we observed. So yes, so that's all from my side. So thank you so much.
Great. Thank you very much, both of you, for extremely interesting talks. We will now open up some questions. So we already have a few questions in the chat already, and I urge you guys, if you have any questions, to just put them in and we'll try and get through as many as possible. But I hope you will forgive me by asking a question for myself first to Joseph. And this is purely just to satisfy my own curiosity, really. I never really considered using zebrafish and zebrafish embryos, particularly for neurological studies. And I wonder, has the zebrafish brain ever been mapped? Like, have they identified regions for motility, for example?
Yeah, that's a really good question. It depends. The answer depends on kind of what level of analysis you mean when you ask about mapping. But in terms of brain regions, the presence of brain regions that serve functions analogous to brain regions in mammals, a lot of that work has been done. So this can get hung up in the fact that early brain development in zebrafish and other teleost fishes, it kind of happens in a different order than it does in mammals. And so structurally, the brain, at a coarse level can look very different than the mammalian brain.
Mhm.
But as the field has delved into, well, what is the forebrain in zebrafish involved in, relative to other brain regions in mice, it's starting to look like that we can identify an analogous structure. So say if you're talking about like the part of the zebrafish brain that is heavily involved in dopamine signal. Well, as the field has progressed, now we can identify that region in zebrafish, and we can say a lot more about what behaviors that brain region regulates and how they relate to the behaviors in other organisms. Does that answer your question?
That's really great. Thank you. Another question here. I see we have an attendee asking that zebrafish, they lack breast tissue and also lack lung tissue. So can we use zebrafish for studying these different types of cancer?
Yes, of course. So as I was showing, it's possible to evaluate different types of solid tumors. It's true that the zebrafish larvae, don't have all these specific tissues as you can have with the mice, but it's possible to create these avatars or these solid primary tumors in the zebrafish larvae. So our clinical pilot studies have shown that even though there is a lack of all these organs, the translatability that we observe is really high. It's an 80% prediction of treatment outcome. So it's just like a container that we use the zebrafish to evaluate response to different treatments, but also as I was showing in the slides, since it's very highly amenable to recapitulate all this the tumor microenvironment, so it can be easily used to study those very early stages of the cancer progression and metastasis.
I think I'll also add to that. The lung is a really good example because it's absolutely correct. Zebrafish don't have lungs, but developmentally and evolutionarily, the zebrafish swim bladder, so the structure that helps them control their position in the water column, is thought to be analogous developmentally to mammalian lungs. And so that tissue, even though it's not exactly a lung, can often be used to model what happens in the mammalian lung.
Okay, I have another question that is in some ways related to that one. So breast cancer cells will predominantly metastasize to bone. So would you predict different results if you perform your experiments at a stage where the fish don't have bones and therefore cannot produce these bone-specific homing signals for the cancer cells? You understand the question?
Yeah, that's true. Yeah, the bone metastasis, we cannot, at the embryo stage, we cannot see that that bone. The embryos don't have bone; they just have some cartilage. So the prediction of bone metastasis, I will say that we can predict like metastasis. The zebrafish embryo can predict metastasis, like in general. It's what we have observed, and progression to the metastatic disease. Yeah, in general. So since they lack in this bone structure, so it's not specifically to bone, but just as a progression tool for the metastatic disease in general. Yeah.
Okay, great. And this is one perhaps you both can answer. Do you think that the lack of an adaptive immune system at the stage at which you use your xenografts could have major effects on the results that you're seeing? So the fact that the embryos don't have an adaptive immune system, would this have maybe a negative or a positive effect, or no effect?
No, well, we can see it from different angles, because if we are seeing that they lack an adaptive immune system, so that works on our advantage when working with xenografts, because we cannot have that rejection. But and probably this is where the question is going. I think more towards the immune oncology, probably. So that the lack of adaptive immune systems, it's not a problem. Well, for the experiments, for our perspective, because we can, as I show, we can inject the human immune cells, lymphocytes, NK cells, PBMCs. So it's possible to have that environment, that human environment in the zebrafish larvae, and evaluate how this the immune response affects the dissemination of cancer cells, and also all their interactions in the tumor microenvironment. This question can have like two different answers depending on the focus.
But a whole conversation, really.
Yeah, I think I'll also add that, like, Gabby's speaking to a way in which, like, the copy paste nature of zebrafish can really in fact be an advantage as a model. But also, of course, depending on the biological question that we're trying to ask, the lack of adaptive immunity early, it develops later, can be an advantage, because it can enable us to separate in our experiment the effects of the innate immune system and the adaptive immune system, which might be harder to do in other models.
Actually, Joseph, you mentioned your different platform indications, but maybe you could describe a bit more on the immune indications.
Yeah. While we're talking about immunology, I think it's worth noting that that is another area where the transparency of the early embryos that both of us were speaking to is a big advantage. And you could see a lot of this in the images that Gabby was sharing, where you can visualize immune cells as they travel through the animal. And we use that to model immune reactions in a variety of tissues. So that could be one example of what we've done in that regard is, and that's commonly done in zebrafish, is a tail wounding assay. So we can wound the zebrafish on their tails and then live image the response of immune cells to that injury. And that's kind of a simple, easy paradigm. But it's also, again, because they're transparent, not just in the tail, but in these early stages in a lot of tissues. That also means that we have access to immune cells in more complex tissues like the brain, where we can then, because of the ease of transgenic manipulation, we can also label immune subtypes. So we can label microglia, for example, and look at microglial responses to different insults.
Okay, another question, perhaps this would be the last one. Do you think it's feasible to use adult zebrafish to conduct biocompatibility of biomedical components despite their small size? Is this something that you guys maybe can comment on, or perhaps you don't work with adults?
I'll start, and Gabby should chime in as well. I think this is a fascinating question, right? Because yeah, we can't. It would be challenging to take a I don't know, like a femur implant and stick it into an adult zebrafish and see if the zebrafish immune system rejects that implant. But because those devices like that or any sort of biomedical component, those are complex components, right? There's chemical additives, surface coatings, all sorts of things that would be much more feasible to test in any stage of zebrafish. So because of the ease of exposing zebrafish to compounds or chemicals, or I think it would be really feasible to check how the animal responds, either at a gross morphological level or even the response of individual tissues or cell types to exposure to a part of a biomedical component.
Okay, great. I'm afraid I think we'll have to leave it there for time reasons. But this has been a really interesting discussion, very interesting talks. I appreciate both of your insight into this topic. I would urge anyone with questions, and I know we couldn't get to every single question, I urge you to email us or to contact us on LinkedIn. We really enjoy hearing from you, and we're very happy to answer any questions you might have. Like I said, this webinar will be uploaded onto our YouTube channel in a few days' time. So please check back, and also you can check our previous webinars. And so that just leaves me to say thank you very much to Dr. Gabriela, and thank you very much, Dr. Joseph. And from all of us here at BioReperia, thank you to watching, and goodbye. Thank you.